Good afternoon to everyone again. We are about to start also our last main talk of the summer school. So it is my honor right now to introduce Dr. Eduard Sabido. Eduard Sabido is the head of the proteomics unit at the Center of Genomic Regulation, the CRG, at University Pompeu Fabra. Dr. Sabido has a PhD in biology from University of Barcelona. He then moved to the Swiss Federal Institute of Technology in Zurich to master then mass spectrometry technologies. His research interests lie in the development of innovative mass spectrometry methods and computational tools for targeting protein quantification, the characterization of protein interactions, and identification of protein post-translational modifications. Dr. Sabido, the stage is yours.
Okay, so thanks, Carlo, for the introduction, and well, welcome everyone. I'm very glad to have and host you here. So this is the second time that we co-organize, as local organizers, the MAX1 in Barcelona, and it has been a pleasure to have you here during this fantastic week in which we learn about high advanced contents and topics on MAX1 and Perseus. So, as Carlo was saying, we are a proteomics unit from the Center for Genomic Regulation and University Pompeu Fabra. So we are located here in Barcelona. So just for reference, so here it's where we are at the moment. So at CCCB, here is where we had dinner on Wednesday, and we are in this building here, which is the Biomedical Research Park in Barcelona, just in the front coast or front line of the city. We are also part of the Spanish national research infrastructure called Omics Tech, and as such, what we do is we provide support for biomedical research through services, collaborative projects, and also through our own research on development of mass spectrometry and proteomics technologies. And in the recent years, we have been working quite extensively on the characterization of histone modifications, characterizing also chromatin and RNA binding proteins. And we have also a genuine interest in the development and implementation of targeted proteomics and data-independent acquisition methods. Beyond the national part, so we are also part of the European proteomics infrastructure, the EPIC Access Consortium, which is a consortium of 18 different institutes around the world, around Europe. And during the last few years, we have already provided access to high-end technology, mass spectrometers, but also most importantly to experts in proteomics for more than 1,500 access dates. We have also put together protocols, like for example, the limited proteolysis protocol that was introduced by Paola yesterday, or for example, some chromatin binding protocols or chromatin binding proteome characterization protocols that we have also developed. So I encourage you to check the website of the consortium to see all the resources that we have there, either protocols or also webinars that we organize in regular basis all along the year. Finally, so our group is also the coordinator of the Protein European Training Network. So this is a network funded by the EU with more than 4 million euros to actually train 15 PhD students that are actually all of them here among the audience. And what they do is actually they do the PhD on developing new machine learning algorithms that will be implemented in softwares like, for example, MAX1 or the MAX1 DIA and many other softwares to better analyze in the future our proteomics data. So what I want to talk today is about the use of linear ion traps in data-independent methods. So DIA or data-independent acquisition methods were already introduced during the week. So on Tuesday, I think it was Jürgen that explained all the new features of MAX1 DIA. And by now, probably you already know that DIA actually uses broad windows for peptide co-fragmentation, right? So this means that if we are interested in scanning peptides, for example, from a mass range of 400 to 1,350, so what we do is we isolate peptides in batches in windows, like for example, 400, 425. We isolate, we co-fragment, and we detect all of them, and then we move a little bit shifted, no, to 425 to 450 and so on and so forth, until we have co-isolated, co-fragmented, and co-detected all the fragment ions derived from the precursors that are in each window. All of the consecutive, the sequential time that we dedicate to all these windows, right? It's a cycle time, so it's a time that we take to do, for example, in this case, one MS1 for the whole mass range and all the series of MS2. And I emphasizing this concept because the cycle time is a concept that will be recurring during the talk. If we take a look at DIA or how DIA works, not so much schematically, but more from the instrument point of view. So what we are doing is using, as you know, hybrid mass spectrometers in which we have different mass analyzers. And we would operate the first mass analyzer, which is normally a quadruple in a broadband filter. So this means that, as I was saying before, we not just filter for one specific M/Z ratio, but we allow many precursors, in this case from 400 to 425, to enter the collision cell. We fragment this, and then we use a second mass analyzer, in this case is in a screening mode, so that we can record what are all these fragments and then generate a very complex MS2. Right, so these are compared to DDA, so these are very complex MS2 spectra that afterwards suffer, like for example, the MAX1 DIA module actually deconvolutes, as it was explained on Tuesday, and then we can extract and assign what fragments corresponds to each of these precursors. So one can repeat this for the different windows, right? And then once we reach, and we have scanned the whole mass range, we start again, right? And again, so this is what we call a cycle, and this is the cycle time, the time that it takes to do all this in a serial mode, in a serial way, until we start again acquiring the data. So currently, DIA relies mostly on high resolution mass analysis, right? So the first mass analyzer, in this case, would be a quadruple, but the second one, right, it's a high resolution mass analyzer, such as the Orbitrap or the time of flight. And then the different flavors that probably many of you are using of DIA methods, like for example, the SWAP method from SAIX, DIA passive from Bruker, DIA or Orbitrap DIA-based DIA from Thermo, or for example, also the Sonar and MSC and other flavors from Waters, all of them relies on this type of high resolution mass spectrometers. So if we see what DIA, or we check how data is generated or acquired in DIA now with a slightly different view, right? So just to compare it to other ways of acquisition, what we can plot here is the M/Z, the retention time, and then what I explained is that just in every cycle, right? So we fragment, we co-isolate, fragment, and detect all of this. So you can see that all these windows are generated independently of where the peptide signals are present, right? So all these ground signals here would be features, right? So as also explained by Jürgen at the beginning of the week. So independently where they are appearing, right? So we would be doing these cycles of co-isolation and co-fragmentation until we cover the whole mass range during the whole retention time. This contrasts, of course, in what we do in DDA, where the fragmentation only happens for visible peptide signals. So we would have the same map, and we would select each of these features for fragmentation until we cover most of them during the whole retention time. And of course, we can miss some of them also during the period. And this also contrasts what we saw in the data acquisition type with what we do in PRM, in which fragmentation only occurs for a list of predefined peptides, right? So in this case, let's say we would be interested in the purple, the blue, and the red one. Then we know the M/Z, and we say, well, let's fragment all the precursors during the whole time that are in these M/Zs, right? And we do this recurrently so that we only have data acquired in these narrow bands. So as a summary, so DIA methods, right? So are deterministic because we know exactly how data will be acquired during the whole run. and this is similar to the PRM or SRM, the classic targeted methods, right? So we know exactly what it will be selected for targeting. It does not depend on the peptide signals, and therefore we say it's deterministic, but it's not targeted, right? So we are not selecting the specific fragments or specific precursors or M over Z values as we would do at PRM. So if we check the different flavors and different methods that have been developed by or with DIA methods, so what we observe is that there are already several methods that have used low resolution linear ion traps to acquire DIA data. And one of them, for example, is this method from Venable and Yates from 2004. There, they split the mass range in windows of 10, and they acquired all the fragmentation ions in the linear ion trap. And they were already showing that for some specific peptides, like these two ones, so the signal to noise ratio in the MS2 was much better than what you would get in the MS1. Similarly, another flavor, which is the Pacific, that was from David Goodlett, was developed and published in Analytical Chemistry in 2009. In this case, they were doing windows of 2.5, and in those days, it took them more than 4 days to actually complete the whole experiment, right? So because the machines were not as high throughput as they are today, and all this data was also generated with linear ion traps. And more recently, the laboratory of Professor Michael McCoss from University of Washington, together with people in Thermo, they have developed the WISIM DIA, in which they use Orbitrap high resolution mass spectrometer for the MS1 with gas phase method that separates different M over Z ranges. And then for each of, in each of these sections, they select or they generate small windows of 12 M over Z, of 12 M over Z that they acquired in the linear trap. And actually, so the use of linear trap in these different methods, in some cases, it was due to that other mass spectrometers, like for example, the Orbitrap was not yet available, whereas in other cases, it was due to was selected in purpose because of the properties of the linear ion trap. And in fact, linear ion trap mass analyzers have one very strong limitation, which is the low mass resolution, mass accuracy and low resolution when we compare it to the Orbitrap and the time of flight. But actually they are very sensitive. They have also trapping capabilities, and they are very fast, right? And then we started thinking, well, is there any application? Is somewhere in the type of experiments that we do where we could get most out of this mass analyzer? And one of the things that came into mind is low input proteomics, right? So single cell proteomics, or when you have hundreds of thousands of cells in which we are very limited in the amount of the proteome that we have. And that we could use these properties, for example, the inherent higher sensitivity or the faster sampling rate to increase our performance in this type of application. And I want to highlight this faster sampling rate because also when we reduce the sample quantity, actually the number of points per peak is also decreases if we keep the sampling rate. And this is important, and we'll see it later. So number of points per peak is not only important for quantification, but also for identification of peptides, because some algorithms actually suffer when they have only 3 points to decide whether this is a peak or a feature or it's not a feature, right? So having this in mind, what we said is, well, let's design an experiment in which we can test all this. And here is where we have the experimental design. So we did the sample preparation with serial dilutions of commercial HeLa from 1 microgram to 1 nanogram. So there are other labs in the world that are doing excellent job in preparing, digesting single cells and small quantities, but we wanted to isolate all this and just concentrate on the acquisition, on the data acquisition method. And then we used an Orbitrap Lumos with combining DIA methods, either acquiring data on the Orbitrap or in the ion trap. And in this case, in this particular case, so we use as data analysis the SpectraNow with the direct DIA approach, which is a library-free approach, but actually you could do this data analysis with any other software, like for example, Diane, or also the Max DIA model that was introduced during the school. So here are the different methods that we tested. So first of all, so we established an MS1 range of 500 to 900 in 40 windows of 10 Dalton each. And then we set up as a standard method or reference method, the DIA method relying on the Orbitrap at resolution 30K with a scan speed of 64 milliseconds and a cycle time of 3.5 seconds. And then we set up 3 different flavors of the DIA method using the ion trap with normal, rapid, and turbo, which are different scanning speeds, and then synchronizing the injection time to the scanning speeds, we reach cycle times of 1.1 to 1.9 seconds, right? And you can see that they are much lower than what we get in the Orbitrap. So we set up this, we set up the methods, we set up the chromatography, and then we went to real analysis. And the first thing I want to show you is actually the overview of the MS1 total ion current, just to show you that, well, for example, when we are in high concentrations, we see, of course, a nice TIC signal, but actually when we go to 1 nanogram, we don't have FAMEs or other ion mobility devices. You can see that this is almost like a blank. So all the signal is dominated by ambient ions, and therefore, actually, this tells us also that, or confirms that DIA methods are especially suited for low input omics, because otherwise it would be tricky to identify precursors in which to rely the fragmentation. And then we went and checked how many peptides we identified for each of the different methods. Here you have a plot of identified precursors, quantities, and in colors the different methods. And what we see here is that Orbitrap-based methods dominate so this log scale, dominate in the upper part, but when we go down in amount, especially below 10 nanograms, actually the DIA IT starts to shine. And among the 3 of different flavors, the one with larger injection times are much better in terms of number of identified precursors. So we can see the same in a different way, right? So if we check the triplicate injections with 1 nanogram, here we have the Orbitrap, the DIA IT normal, right? Which more than doubles the number of identifications, and then the rapid one, but and finally with the turbo, we don't identify anything. It goes too fast and it accumulates very few ions, and we don't have enough signal. So what we see is that ion trap DIA methods identify more precursors than, for example, what we rely with Orbitrap based DIA based methods in low input omics. But things are not just about number of identified proteins. We wanted also to see how well was the sequence coverage, right? So how confident we were in the sequence coverage. And here what you see is that actually we check the distribution of peaks, fragments per peak, right? So how many Y and B ions we have for each of the identified peptides. And what we see here is that with 1 microgram, right? So in red Orbitrap, in blue ion trap, this will be recurrent during the presentation. Well, the distribution is around 10 fragments per peak, whereas, for example, this suffers very much in the Orbitrap when we go down to 10 nanograms, and then the coverage that we get per each sequence, it's much poor with the relying on the detection of the ion fragments in the Orbitrap than what we do in the ion trap. Right, so I showed you, right, so that we are able, by using ion trap-based DIA, that we can identify more peptides and have more sequence coverage when working with low inputs. Right? The thing is, well, is this due to the intrinsic sensitivity, or it's just by going faster, right? And then you can see that, for example, we have cycle times of around 1 compared to the 3 seconds of the Orbitrap, and then we said, well, maybe if we operate the mass spectrometer faster, so for example, lower resolution with the Orbitrap, we would get the same results, right? And it's just a matter of cycle time. But then what we did is we repeat the experiment by, instead of using Orbitrap 30K, using also 15K and 7.5K. And what you see here is that actually increasing the scanning rates with the Orbitrap that doesn't improve the peptide identification, right? So in red you have the reference method, and then in the other two colors reducing the resolution and also the injection time. So this drop in performance is probably due to the combination of both, right? Reducing the resolution and using smaller injection times that do not compensate for this faster speed. So operating the Orbitrap in low resolution and faster scanning rates is not going to achieve, or it's not achieving the same results as we get with the ion trap. So in general, right, in these first few slides, what I showed is that actually ion trap DIA methods outperform DIA Orbitrap in low input proteomics. But sometimes, and this was also introduced by Albert with the histone phosphorylation, right? So software have some expectations, right? And when you start doing methods that are not as the mainstream, you might incur in error. So we started thinking, well, are these peptide identifications correct? So, right? So it's not just about having more, but we want to know that these are correct. And then we started looking manually for the spectra that we were identified, right? And here, for example, I show you in blue the experimental spectra for this particular peptide with the DIA ion trap using 10 nanograms, the DIA Orbitrap using 1 microgram. You can see that the experimental ones match each other. And then we also use one of these prediction tools, like, for example, the one that was also introduced here during the week to predict what we would expect, right? And what we see is that both Orbitrap with this is the reference and normal operating in 1 microgram, it matches very well, but also it does the one that we get, the identification that we get with the DIA ion trap based method. We did this for another peptide, right? Same results, but of course we are not able to check manually one by one everything, and we wanted to have also some global parameters that could allow us to be confident on what we're seeing, right? And then we started checking what was the overlap among the different methods. So you can see, or you can interpret these bars as Venn diagrams, and we will start with the 1 microgram input. Here what you see is that in red would be the circle of what are the precursors identified with the Orbitrap, and in blue, what are the ones identified with the ion trap. Gray is the overlap between the two methods, right? And what you can see here is that, as we saw at the beginning, using the Orbitrap, we identify many more peptides than what we do with the ion trap, and that actually we include almost most of them. But when we go to low input, the situation is exactly the opposite, right? So we identify some precursors with the Orbitrap, but all of them are recapitulated by the ion trap, that not only recapitulates what we see with the Orbitrap, but also gains in number of identifications. And if we check what are the distribution of abundance of the peptides that we identify, so here, for example, in the Orbitrap, in dark red, what we see is a distribution of all the precursors that have been identified with low quantity, with high quantities, and then what we ask here is what are, or which is the abundance of the ones that we identify with low abundance, right? And what we see is that they are among the most abundant one in the sample, which is what you expect, right, when you go to low quantities. And the same behavior happens with the ion trap. And this is good because if this would be random identifications or the false discovery rate would be not controlled, this would be randomly distributed among this distribution. And finally, we also check the retention time. Everything that we see matches the retention time of what we would expect. And here what we compared is the DIA-IT with high concentration and for high amount of sample and low amount of sample. We see that all match very well. Also the Orbitrap, we made the same comparison with the Orbitrap, and then the ion trap against the Orbitrap. And what we see is that all the identified peptides match the expected retention time. So in general, what I'm saying here is that not only we get more identifications, more sequence coverage, but that we are confident on what we are seeing. As I said before, points per peak are important, not only for the identification, right? So because if we have more points per peak or less points, maybe the algorithms have difficulties of knowing whether this triangular peak is actually a feature or not, but also they are important for quantification. And this is something that also we wanted to assess in these linear ion trap methods, right? And the number of points per peak are important for quantification because, as you know, the more points that we have, the better it is to reconstruct the original peak here in red. If we have less points, and there because of the cycle time, then, for example, given two identical peaks in red here, depending on where these points, sampling points lay in the peak, the area that we recover or that we reconstruct might be different. So what we expect is that if we are in these type of situations, coefficients of variation would be higher than what we would be in here. And this is what we check, right? So first of all, how many points per peak we had in both methods. So the orbit trap in red, DIA ion trap in blue. And we see that, of course, due to the faster cycle time, points per peak are always higher in the DIA-IT method, both in high and low quantities, but that then this has an impact or a higher impact in CVs on how good or how bad are these CVs, especially in the low quantities and 10 nanograms, right? And you can see that distributions of CVs actually are much worse with the DIA orbit trap than what we get with the DIA ion trap. We also correlate the abundances, right? So actually that we got from the ion trap and the orbit trap, the absolute numbers are not comparable because the detectors are different, but actually we wanted to see whether there was a correlation here and whether this was well ranked. And what we see is that the correlation that we get at 10 nanograms is actually similar or equivalent to the one that we get at 1 microgram. And also, for example, recently, the laboratory of Professor Michael McCoss has also shown that actually in PRM, the use of ion trap methods with, if you select for the good transitions manually, so have better quantification properties that what you would get from orbit trap. And then one can think that what has been described by McCoss for PRM, one could apply it in high throughput for DIA ion trap based methods with algorithms that actually automatically detects interferences, like for example, Avantgarde from Sebastian Baca and Steve Carr, that could actually refine all the data and get much better quantification properties of the ones that I'm showing here. So as a first conclusion, the use of ion traps for analysis of MS2 in DIA methods is well suited for low input omics. And when we talk about low input omics, I'm referring to equal or less than 10 nanograms. And I showed you that they render more identified precursors, better sequence coverage. They also have better cycle times, and therefore they originate more points per peak. And this actually, or consequently gives better quantification properties. If we go back to our method overview, right, and we already saw, and we already saw before, and we take a look at the cycle times, we see that actually we are almost or over 3 times faster with the ion trap than in the orbit trap, right? And then also we started thinking, well, what we can do with this spare time? Can we invest some of this cycle time to do other things, right? And to even improve our methods. And the first thing that we thought is, well, we could gain a specificity because we could dedicate some of the cycle time, but to use narrower isolation windows. So then what we did is we started checking also another method that instead of using 40 windows of 10 Daltons was using 10 100 windows of 4 Daltons, because narrower windows actually reduce the complexity of the fragmentation spectra and therefore increase the specificity and the sensitivity of the method. And when we check this with exactly the same type of input sample, what we see is that actually narrow isolation windows with DIA-IT boost peptide identifications also in low input proteomics. And you can see here that again, in 1 microgram, the clear dominating method is the Orbitrap. But actually when we go to low input proteomics, so the ion trap is better, but then if we use narrow isolation windows, we can even boost this number of peptide identifications. And actually we tested this not only for this particular case, but also for all the different flavors of methods based on DIA-IT that we were checking. And here you have in red the one with higher number of windows, in blue the one with 40 windows, and you can see that regardless of the method, the red one is always higher than the blue one, right? So that all methods actually benefit from the use of these narrower isolation windows. Not only this, but actually, if we check again coefficient of variation of this method, what we can see is that actually the coefficient of variation of this new method actually is maintained despite the increase of the cycle time, right? So that it's not yet in the critical zone of cycle time that would actually increase the coefficient of variation. So I showed you now one of the applications on how to use this spare cycle time. Another way to spare the cycle time is actually using ion mobility, for instance, in which we would also gain a specificity. And we were thinking, well, we could combine this DIA ion trap with ion mobility. And now I want to show you some data that has appeared also this in a bioarchive paper from Professor Erwin Schulf from Denmark, in which actually they did a very similar experiment. And what they did is they took this type of DIA ion trap based method, they reproduced the results that we had, but also what they tried then is to introduce this FAMES interface, right, in which they filter the several single ion charge. And by doing this, actually what they can increase not only a little bit, but multiply the number of peptide identifications that we get, right? So they tested many different quantities that we did, but also in turbo, rapid, and normal. And what they see is that actually introducing this FAMES filter, because actually this is a cleaning step, could actually benefit much more the specificity of these ion trap based methods. So now what we are also trying is actually to use, together with Thermo, this model to introduce compensation voltages, so not just one, but actually combining maybe two to see whether this could increase even more the specificity that was described in this paper. And then I also want to I took this screenshot from the bioarchive paper, because one thing that they did also is they analyzed the same data with two versions, in this case of Spectronaut. Right, and what you can see is that when they use Spectronaut 15 and 16, actually the number of identifications increased. And also we saw this during this week, is that software that analyzes DIA actually is improving very fast, right? And many machine learning algorithms are currently at the moment being developed that have been or will be introduced in these softwares, like for example, the new features of MaxQuant DIA. And therefore, right, it's important, well, it's a very increasing or a very evolving field in which I think that, for example, data from the DIA-IT methods that I'm showing could benefit even more with all the machine learning algorithms that would predict not only retention time, fragmentation, but also ion mobility. Then, in another way, or I showed you how to gain a specificity in two different ways. So reducing the window size and also the introduction of ion mobility. But another thing that one could think is actually we could gain time, right? So if we have spared cycle time, we could make it shorter gradients, right? And this would be very good for high throughput applications, like, for example, the analysis of plasma samples, right? Big cohorts. Let's think about hundreds or thousands of samples. And then we tested this also. And here we tested it with 100 nanogram of plasma sample with short gradients of only 30 minutes. This is quite, well, it's not only a complex sample, but abundant sample. And of course, the DIA-OT dominates in terms of number of precursors. Here, among the DIA-IT, the ion trap methods, the turbo one, actually it's performing a little bit better than the normal, but it does not reach the number of precursors that we get with the Orbitrap. But if we dive into the data, to the raw data, and we look, for example, again, the total ion current, and we see, for example, here I just plot the total ion current that we get with the Orbitrap with cycles of, in this case, we used 15 resolution, which is, if you see many publications of high throughput plasma proteomics, this is the standard method that is currently being used by different labs with a cycle time of 2.35 seconds. And this is again, the total ion current of the DIA ion trap of 40 windows of 10 Daltons each in turbo mode, right? With a cycle of 1.27. And one would say, well, they are very similar. With the Orbitrap, we get much more identification, so let's go for the Orbitrap. But when we look to specific peptides, what we see are these type of transitions, right? These are the elution profiles of the fragment ions. And what I see here is that actually we have triangular peptides with this cycle time of 2.35, and actually much better defined peaks with the linear ion trap based method. So peptide identifications are not enough. We also want good, accurate, and precise quantification. And I can show you, for example, another peptide, and we also see these triangular peptides that we get with the DIA Orbitrap method compared to the DIA, right? But again, as with the spectra, we cannot check for everything or all the peaks here, and I could also cherry pick some good and bad examples. So therefore, what we said is, well, let's see at the distribution of the number of peaks, what we get with one and another method. And what we see is that with the Orbitrap, we have a median of 3.8 points per peak, right? Just remember that 3 is triangular, right? And whereas using the ion trap, we get or almost double the number of points per peak, and therefore better quantification properties. So with this, right, so I conclude the second part on how to use this spare time, right? So with ion trap, we go faster, and then we can use part of this spare time to either gain specificity by using narrow windows or introducing new analytical techniques, like, for example, ion mobility, or to reduce time in data acquisition for high throughput applications. And then before closing, I also want to have some words on quality control, right? So what I showed today is a lot of an analytical project, and therefore we want to be sure and confident that our instruments, our systems work as they are expected to work. So we dedicate and invest quite a lot of time on analyzing quality control samples between each of the samples. So each replicate that I showed data for in the previous slide, it's controlled with its respective quality control sample. And therefore, we have quite a lot of confidence on the data that we generate. Of course, this has also brings some new problems, that it duplicates all this quality control data needs to be analyzed, right? And then it generates quite a lot of work. So what we have done in the last 5 years is actually develop what we call the QCloud pipeline that automatically processes all the quality control samples. So what we have is that automatically the system with a small applet that we have in the acquisition computer actually uploads all the quality control samples into an FTP server, and there with Nextflow, which is a workflow language that was developed at the Center for Genomic Regulation, and also the models of OpenMS from the Tübingen University. So we are actually automatically analyzing and extracting all the QC parameters that are afterwards displayed in the web. And you can see here how we control our mass spectrometers. So these are the mass spectrometers in our unit that are not actually now being registered here. And you can see how we can actually see the performance for many different quality control measurements. And in the last 5 years, we have processed over 100,000 quality control samples from 24 different laboratories and 61 different instruments. So also we think that science is a collaborative effort. So what we did is we reshape all the QCloud system so that it's open to all the different platforms and mass spectrometry proteomics laboratories in the world. And now one can access, and I actually invite you to check for this URL and register your own and use also the automatic pipelines and have this type of website interface for your own mass spectrometers. And with this, I would like to thank all the members of the proteomics unit, right? So Cristina, Eva Guadalupe, Roger, Zara, Olga, Enrique, and Selena, who are most of them also in the audience today, our institutions, the Center for Genomic Regulation, University Pompeu Fabra, and of course, all our funders. And of course, all of you for your attention. Thank you very much.
Any questions? Yes.
Thanks for the great talk. So the quantification you showed, this is from MS1 level, or is this from the transitions?
So this is from MS2.
Okay.
Yes.
And
sometimes also Spectronaut combines MS1 and MS2.
Yeah.
Right?
And did you also do, I mean, for judging if the quantification like these 2-3 species mixtures? Because I think in the CV, you don't really see the problem of overlapping transitions very well, right? Because it's the same for all in the sample, basically. But one would see it much better in like these 2 species mixtures, right? Did you try any of these?
So you mean controlled mixes?
Yeah.
No, so we didn't do or went into this yet, but it's something of course something to check. Of course.
Thank you.
Great talk. A small technical question. Did you in the plasma proteome thing, did you deplete the samples? And if yes, how?
Okay, good. So we don't deplete the samples, and we go for, or we try to prioritize single shot plasma injections without depleting them.
Hi, good talk. Thank you. I have a question. When you compare like the 4 methods, like 3 with IT and 1 method for, with Orbitra, did you took into account to get like to design some windows that way that you will have the same number of precursors in each window to compare between these 4 conditions, or was that Orbitra method developed like from before and just compare these 4 conditions?
No, so the sample and the isolation scheme is exactly the same. So how the different precursors, or what are the precursors and which lay in each window, therefore are also the same. It's only about speed and injection time that we are changing. Well, of course, and mass analyzer. Is it clear? Or
I think that when you want to compare this method,
yes,
I would tend to think that, okay, I should design like in the Orbit app, a method that will get exactly, no, approximately the same number of precursors in each window. So I don't know if 40 windows will be the condition to keep. Maybe you change a little bit the fill time and the cycle time to reach these conditions.
okay. I don't know whether I understand, but I answer 2 things, and maybe one is the correct one. So first of all, so the isolation scheme are the same, or is the same for all the methods that we we tested, and also the sample. Therefore, how the or which peptides lay in each window are exactly the same and totally comparable among methods. Another thing is whether we could have used different window sizes according to the complexity of each mass range, that we didn't. So we always use these 40 windows of 10 Daltons. Other laboratories have explored what would happen to use variable window size depending on the complexity of the sample.